Oh, I can't hear you. Where's everybody else? You recording on? Yeah, he's recording. Okay. I wonder if I can hear you. Okay, it's not saying. Wait, are you? T-shirt. T-shirt. They're gonna confirm some bios in class. Yeah. Do you have to have a camera? I don't think so. Because you might. No. Oh. Gosh. Just so much better in person.
All right, class, let's get going. Um, as you can tell, my voice is a little rough, but I didn't bring my microphone with me, so it's important if you start talking quietly, if you can't hear me, just unmute yourself and say something about it. But normally when I start, it'll be loud enough. Um, so there's an echo. I can't, doesn't make sense. I'm just using the laptop microphone. Let me go to audio. Okay, let me see. I think the echo has gone away. I switched the microphone speakers. Yep, seems good. So first thing to tell you is that uh, I tested positive for COVID on Tuesday. I had tested negative on Sunday, and Friday and Wednesday we had some COVID in our lab, so I was checking all along. So I think it's kind of rare, but it does happen. And so I'm glad I was wearing a mask. But ooh, yeah, be sure to mute your yeah. So, anyways, if I were you, 200 people out there, it might not be a bad idea to test in a couple of days and watch for symptoms. But hopefully the mask worked, and I didn't really have a lot of communication with students. So I've got my fingers crossed. Any questions? Let's see, there is something in chat. Let me just check that. Okay, good. Let's see. So might be a little bit of a delay, but my plan is to post the grades on Friday, probably evening. I'm expecting to get into work Friday and get the exams scanned since I'm feeling a lot better. And that will be already 6 days from my first symptoms. But that's when you can expect your exam grades. Already posted, I think, are your extra credit points, and also posted are the keys to the exam. What will happen is not probably not Friday, but maybe Monday, and two or 3 days, I'll bring copies of the Scantrons in. They'll be separated by what version. So try to recall what version you took, A, B, C, or D. And then it's kind of a scrum to find your Scantron in with everybody else's. But I'll bring them in before class and also stay a few minutes after class with them. And I'll do that on a few days. But it's not going to be like forever that I keep bringing them in. So you want to have a little time before class or after class. Let's just say Monday, Wednesday, Friday of next week. And then after that, you, you will just have to wait till the next exam to get a Scantron. Okay, any questions about that? Okay, so we were talking about where proteins are made in the cell, 2 places, and also how they get from where they're made to where they're functional, since you have a lot of different proteins doing different jobs in different parts of the cell. And the first example of that is a protein that's made in the or proteins that are made in the cytosol and that move to the nucleus. So any kind of nuclear protein, like a histone protein, condensins, all those proteins we talked about when we were talking about chromatin structure, they all have to be inside the nucleus. And so they're made in the cytosol, they have to get
It's
in.
already in the workflow document.
Whoops. Mute, please mute.
You're
I
not,
wonder if I can
this is, you
mute.
asked how to
I bet I can.
assign for,
It'd be worth just waiting a minute to see if I can. Pause. There should be a way for me to mute everybody, but I haven't found it. So we may have that same problem again. Okay. Let's go back up here. So just because it's been a minute before we talked about it, remember there's 3 sort of components as far as proteins getting into the nucleus. Every protein that gets into the nucleus has a nuclear localization signal, lyse and ARG-rich amino acid sequence that is at the N terminus. So proteins get not just the function, the part that does the function, whatever that is, but they also have an address. And a nuclear localization signal is an example of an address that gets the protein from the cytosol to the nucleus. And how it does that is through the second component of import. It's really a duo here between important and RAN GTP. Important is actually binds to the nuclear localization signal, represented here by this little triangle, and pulls the already folded protein into the nucleus through the nuclear pore. And then this is where RAN comes in, because you can't have all your importance stuck in the nucleus. You want it out in the cytosol where it can pull in more cargo. So RAN is what pushes the important back out into the cytosol. And that involves hydrolysis. And RAN also is what dissociates the important from the cargo. And RAN hydrolysis is, besides pushing it through, is also what dissociates RAN from the important let you have free important that can then pick up more cargo. Any questions about that? Okay, so that's just the nuclear pore, as we mentioned, is this large structure that allows proteins, even protein complexes, to get through, and it's asymmetric. It looks different on the N-terminal side than the C-terminal side, because on the N-terminal side, you get this nuclear basket, which you can see in the cartoon form here, or in this transmission EM. And so that's on the nuclear side here. Here's the cytosolic side. It also has nuclear fibrils, but they're not all tied together, so you don't really see them as much in the in this thin section. Okay, so I think that's all we want to say about the nucleus. Any questions about that before we move on? So another place that cytosolic proteins can go is the peroxisome. We haven't really talked a lot about the peroxisome, but it is an important organelle for metabolism. So it's a standard organelle with a lipid bilayer. We saw some pictures of this earlier. Sometimes what you can see is a dark core within this, and this is almost crystallized enzymes. It's so dense with metabolic enzymes that they pack into almost like a crystal structure that's staining darkly. That's called the crystallized core. So it's spherical thing, kind of nearly the size of the peroxisome is when we looked at different ones. Sorry, nearly the size of the mitochondria. I don't see any mitochondria here, but what it does is it breaks down these long chain fatty acids. We talked last week about how you synthesize fatty acids in the liver and then move it into other tissue. But one of the main roles of these fatty acids, besides serving in the bilayer, is for energy. And so, so a lot of that is happening in the peroxisome. Another job the peroxisome has is to remove toxic molecules. And it's present in most cell types. So that's the peroxisome. How do you get proteins into it? Well, what you'll recognize is it's a very kind of similar type mechanism, but with differences for each one of these organelles that we're importing into. The nucleus, the peroxisome, and then the mitochondria we'll talk about in a minute. You always have a signal. And for the peroxisome, it's called PTS, peroxisomal targeting signal. And there actually are two types of signals in two different classes of proteins that go into the peroxisome. So one is called the PTS1 signal, and it's shown here in magenta. And so it's, again, you have the folded functional protein, and you have the signal. Now, instead of important binding to it, in the PTS1 pathway, PEX5 binds to it. And that's on the C-terminus, which is different than the nuclear localization signal, which is almost always on the N-terminus. However, there's another signal you can have. You don't need both of them, just one or the other. It's on the N-terminus, and that's in the PTS2 pathway. So that's the PTS2 signal, and it binds to, and it binds to PEC7. So these are then cytosolic receptor proteins, as they're called, that work like important. In other words, they move their cargo into the peroxisome, and then they are able to, they are able to release it and then move back to the cytosol. So they don't really have the equivalent of RAN, GTP, GDP, at least it's not known that there's another protein that moves them back. It is known the structure, though, that they go through. It's somewhat smaller than the nuclear pore complex. You can't bring in huge complexes of proteins, but you can bring in a fully folded protein, and that's called the docking ring complex. Sometimes you call all these things like the docking ring complex and the nuclear pore importomers, which I never really liked that term, but they're all doing kind of the same thing, but maybe in a slightly different way. And so the key is then that for proteins that go into the peroxisome, they have this PTS1 or PTS2 signal. For ones that are going into the nucleus, they have an NLS signal. So that's how everything gets sorted out. Oops. So let's do this question. I think it'll work because I started it. Okay, let me close that a minute. Go back to this. Countdown. 5, 4, 3, 2, 1. Hopefully this is not going to give me an. Yeah, most of you got the right answer, number C. Let's go back to that PowerPoint. These are these nuclear fibrils forming a nuclear basket. So that means that either C or E must be correct. And so this is where the nuclear basket is, that's the nuclear side. So this must be the cytosolic side. Any questions about that? Okay, so here's the last set of proteins that are, let me go back to a full screen. Here's the last set of proteins that we'll talk about that are made in the cytosol and imported somewhere else. By the way, don't forget that the fourth place that cytosol proteins can go to when they're made in the cytosol is to stay in the cytosol. There's a lot of proteins in the cytosol, and those just have no localization signal at all. So it's the absence of localization signals that leads a cytosolic protein to stay there. So these that are going into the mitochondria, it's kind of the same old story. They have a target sequence, but it's now, instead of PTS1 or 2 or NLS, it's called MTS. And it's also recognized, but it's a little bit different than the previous two cases, because where it's recognized is actually right on this protein complex called the Tim and Tom channel. And recognize the one on the outer membrane is called Tom for outer membrane, and the inner membrane one is called Tim. And recall that the mitochondria, unlike a lot of membranes, is got a double membrane. It's not just that it has a lipid bilayer, it has two lipid bilayers. And so the proteins that have to go in have to actually get through both membranes. And they do that by this large complex. Each one of these is many proteins that passes the protein first from the cytosol into the intermembrane space, that's Tom, and then from the intermembrane space into the mitochondrial matrix, which is the inside of the mitochondria. So that's how it works. It's kind of different, and it doesn't have an important kind of a protein. It just binds directly to the receptor that is on this surface of the mitochondria. It's another big difference is that with this from the previous two is that these proteins are not folded proteins like the ones that go into the nucleus or into the peroxisome. They are synthesized, and a protein called a chaperone binds to them and keeps them in the unfolded state. And an example of that is HSP70, shown here as this purple bean-shaped thing. So in order for the proteins to get into the mitochondria, they kind of snake through this Tim-Tom channel. Once they get to the, oops, I want to move this somewhere else. Once they get to the mitochondrial matrix, that another version of HSP70, not the same proteins that are in the cytosol, but a mitochondrial form of this HSP70 now helps it to fold up. And another thing that's happening is that while the protein is folded or around the same time, using the energy of ATP, this MTS sequence is cleaved off. The nuclear localization signal and PEX1 or PTS1 and PTS2 they are not cleaved off. So you can see it's sort of the same story, but with little differences each time. One other thing on this slide, two other things. One, we're not going to discuss chloroplast important in plants, but it turns out it's very similar to what you see with mitochondria. And second, we really haven't talked a lot about the mitochondria yet, but it's important to say what those functions are. As you may know, the mitochondria is there to generate energy, not so much from fatty acids, that's the peroxisome's job, but from all other kinds of nutrients, carbohydrates, etc. It generates high energy molecules like ATP. It has a second sort of unrelated job. It regulates reactive oxygen species levels. It can produce those ROS, as they're sometimes called, reactive oxygen species, but it can also remove them. So they're important signals in the cell, but at high levels, they are toxic. And partly related to that, the mitochondria plays some key roles in programmed cell death, which we'll talk about later in the class. Any questions about this so far? Just be sure if you have questions to unmute yourself first.
I think I had a question, but I put it in the chat. Do you want us to just ask it to you directly, or?
Let me just see. For some reason, when I have it this way, the chat is not showing up. Let me see if I can see it. Nope. Don't know why. Yeah, just ask me directly. That's probably faster.
Okay, my question was, is there a difference between chaperone HSP70 and mitochondrial HSP70 that we might need to know for the exam, other than the fact that chaperone is on the cytosolic side and mitochondrial is in the matrix?
Yeah, that is kind of the main difference. But another difference is that this chaperone here is keeping the cytosolic one is keeping the protein unfolded, while the mitochondrial one, its job is to fold the protein once it gets through. Any other questions, Quinn, in the chat, like from other people? Or was that the only one?
I'm not seeing any.
Okay. All right. Thanks. Yeah, it's probably easier to just ask me in the future, because for whatever reason, that chat thing is not showing up for me. Okay, here's a video.
Mitochondria are organ.
And now we can move to the other direction that the other place that proteins are made in the cell, which is on the surface of the ER. So the surface of the ER is covered with ribosomes, and that is kind of a different set of ribosomes than the set that is continue to translate proteins in the cytosol. Although, as you'll see, they come from the same source. But the proteins that are made on the surface of the ER are proteins that go into the ER. And also, this is probably most of the membrane proteins elsewhere in the cell, like the plasma membrane or the Golgi. A lot of those are synthesized on the ER. And so it's a very extensive membrane. It forms kind of a web throughout the whole cell. You can see this kind of layered structure here, and that's the membrane, and these small dots then are the ribosomes that are covering that. So you don't really see the smooth ER here. It's more distal from the nucleus, which is here. But you see the ER. My dog just walked into my lecture, turned around and walked out again. So that's what you get when you're a dog. You can do that. So let's see.
This man's talking about his damn dog right now.
Yeah.
Yeah, because my dog just walked into my
You're,
lecture and then walked out. Guess you guys
you're
kind of want to
not-
be that dog, huh?
You're
Because then
not,
people keep leaving the Zoom.
you're
Hey,
not
Kirsten,
muted,
you're unmuted.
Kirsten. Yes. Yeah, go ahead. Mute. Yes, you and I have the same sense of humor. So so let's see. ER is not so yeah, so you can't see the smooth ER where all the lipids are made, but you can see how extensive the rough ER is. That's proximal or near to actually contiguous with the nuclear membrane. And yes, it says here is 30 times more ER membrane than plasma membrane. It extends in this web that in a lot of conditions goes right from the nucleus all the way up to the plasma membrane. And that's shown here when an ER protein is tagged with a green fluorescent protein. So you light up the whole, I think it's the ER, right? Probably on the ER membrane, but what you're seeing here is essentially this network of very thin vesicle, that very thin kind of web-like vesicle. So if you look at one particular part here, it's got a membrane on this side covered with ribosomes and a membrane on this side covered with ribosomes. And even though it's thin, there's an inside, which is called the ER lumen. So proteins that are made on the ER membrane, some of them go inside into the lumen, some of them stay as transmembrane proteins. And that's what we'll talk about here. Let's see. So the interesting thing is that all these ribosomes that yeah, I think we'll talk about this in a minute. Let's just talk about the ribosomes that are attached to the ER. So here's the ER membrane, and then in gold we have the ribosomes. The ribosomes are already attached to the mRNA before they reach the ER. So what coats the ER membrane with ribosomes is that ribosomes connected with mRNA and starting to translate it move to the ER. And how do those mRNAs and ribosomes know to get there? Well, it's the same kind of thing. It has a signal sequence called the signal peptide that is that is on the N-terminus. And so as this ribosome begins to synthesize a new protein, which is shown here, the first thing it synthesizes is this signal peptide. And that signal peptide If we look at another ribosome that it's gone a little bit farther, it gets recognized by this complex called the signal recognition particle, shown here in the blue. And it's that signal recognition particle that actually takes the whole ribosome, new protein or beginning protein, and even the RNA is all pulled along into to the ER membrane by the interaction between the SRP and the SRP receptor. And that's what really has moved this here. So you'll see that once this, let's talk about this, once this finds a free SRP receptor, it'll also bind to the ER. Once this picks up a signal recognition particle and that particle then finds an SRP receptor, it will also come and bind here. So you can see all those ribosomes are actually active ribosomes, and they don't really get to the ER until they start translating, because the very first thing you need is the signal peptide. So the SRP receptor then is connected to the protein translocator complex, which also serves as a second receptor for the ribosome. You kind of have this for the nascent protein, but you're also binding to the ribosome. And most importantly, this protein translocator allows the protein to move through the through this channel and into the cell. And one thing that's very interesting is what pushes the protein through the ER membrane is actually the energy or the force created by translation. As the ribosome is making the polypeptide chain one amino acid longer each time, that force is actually pushing that new polypeptide all the way through until you get a complete folded protein inside the lumen. So this is what happens when you have an ER protein that you want in the lumen. You call this sometimes a soluble protein, as opposed to a protein that's on the that is a membrane protein, which we'll talk about in a minute. One last thing I didn't mention before is that at an early stage, as the protein is coming through, it's important to clip the signal peptide, which is still connected with other parts of the complex, and that lets the protein go in. And the enzyme that does that is the signal peptidase. Here, what they point out is that it's really as the ribosome is kind of passed on to the protein translocator complex, that the SRP is liberated, but you don't really get that protein free to get inside the cell until that signal sequence is removed.
Do we need to know what causes the signal sequence to be removed?
You just need to know that it's signal peptidase. So signal peptidase, to describe that a little bit more, is a kind of a protease, which is able to make this clip or break in the growing protein. So this, just like the signal peptide is recognized by the SRP, which binds it, the same sequence is recognized by the signal peptidase, which cleaves it. And then this protein, this little piece of protein is just degraded. Any other questions about this? So you can see it's a multi-step process. The bottom line is you want to get proteins with this signal peptide sequence into the lumen, though part of that is removing that signal peptide sequence. So another thing is the whole issue of these signal sequences. This is true for both the ones that are getting into the ER that we just talked about and the ones that we talked about before that are from the cytosol. How do you really know that that sequence is important? Well, it turns out you can do the test. You can take a ER signal sequence and put it on a cytosolic protein and say you want to do 2 experiments. You can remove it from the ER protein that is normally there. So usually it's a genetic manipulation that you would do to change the gene that makes that ER protein so it doesn't have a signal sequence, and change the gene that makes the cytosol protein so that it does have that sequence. And sure enough, what you get is a swap. No sequence. It doesn't matter that this is an ER protein. It stays in the cytosol. And this cytosolic protein, just having that signal peptide is enough to get it into the ER. And you can play this game not just with ER proteins, but mitochondrial proteins or peroxisomal proteins. Wherever you get a localization signal, you find that it is both necessary and sufficient for localization. Well, as an information molecule, it's sufficient. Of course, you need a lot of enzymes besides this signal sequence that recognize it and that pull it through. Let's see. So I kind of alluded to this before. Maybe I should have talked about it before, but it turns out that all proteins, whether they're going to the ER or they're cytosolic proteins, they start out in the cytosol. So here's a protein with no ER signal sequence. Maybe it has a nuclear localization sequence or whatever, but it just doesn't have an ER signal sequence. It's going to stay in this cytosol. So this is the new protein being made in blue, and here's the green ribosome. Here's the transcript in blue. Here's the ribosome, and then here's the little new protein coming out, just getting larger. But it doesn't have an ER signal sequence, so it's going to stay there. On the other hand, if it did have an ER signal sequence, still start in the same place in the cytosol, but now it's going to be attracted to the ER thanks to the SRP and the SRP receptor and the protein translocator. But then what's interesting is they're not really 2 pools, they're a common pool. Once these ribosomes are finished, they go back. They may end up being an ER ribosome with their next transcript, or they may be a cytosolic ribosome. Any questions about that? Okay, so as I promised, not all the proteins that go into the ER that are synthesized on the surface of the ER end up in the ER lumen. Some end up in the cytosol. Some end up as transmembrane proteins. And there can be some complex topologies of proteins that are transmembrane proteins. We looked at a few of them, but they cross the protein multiple times. But what we want to just talk about here is the single pass proteins, meaning it has just one transmembrane domain. It has a extracellular domain and an intracellular domain that's in the lumen. So this extracellular, you shouldn't call it extracellular. It's got a cytosolic domain and an ER lumen domain, and it has a transmembrane domain. Well, how do you do this? In other words, instead of the protein translocator just the whole protein getting pushed through by the ribosome, now it has to stop. And once again, this is done because of a sequence that's encoded in the gene called a stop transfer sequence. And this is usually a very hydrophobic sequence, so it's going to be great for the transmembrane domain and all those fatty acids that are in the core of the ER membrane. So basically, what happens is it starts the same. You have a start transfer sequence that is basically like a signal peptide. You can actually have start transfer sequences for more complex topology that are not N-terminal. But the one we talked about before, it's called the signal peptide, is what we're talking about here. But it's part of a broader class called the start transfer sequence because it's starting the transfer. And you get the signal peptidase cutting it up. Everything is the same, except that when this hydrophobic region gets to the middle, the translocation stops. And the ribosome dissociates. So part of that is just it's a very hydrophobic sequence, and it stops when it gets into a very hydrophobic environment. And there's also more going on than that. But for this class, all you need to know is that when you get to that hydrophobic stop transfer sequence, then you stop transporting the protein or translocating the protein into the ER. And although the ribosomes are not shown here and all the other things, all of that is exactly the same. So you still have this sequence recognized by the SRP, brought to the SRP receptor. It's still clipped. It's still driven through by the force of the ribosome. Any questions about that? Okay, that seems like a good slide to stop on. I'll post an announcement seeing whether I'll be able to lecture in person on Friday or if it'll still be recorded Zoom. So I'll let you know. All right. See you Friday in one way or the other.
Hey, real quick, there's a question in chat. Can we
Oh
answer that
yeah,
real quick?
I can. Let me. I can see them now.
Okay.
Let's see. Is there a difference between you? I've got that one yet. Yes. Jingyu for the signal is unattached to stop the growing train. Yes. No, actually, when the signal peptide is unattached, it allows the chain to keep growing. So it can't really, the signal peptide never gets fully, let me go back into that. Signal peptide never really gets fully into the cell. And it is also usually hydrophobic. And so it has to be clipped off before the peptide can get in. Does that answer your question if you're still there? Okay, I think that was the only question. Oh, what stops the chain from growing? There is one thing. The only thing that stops the chain from growing is that we've talked about is the hydrophobic. It's not really. I don't know if I said that it stops it from growing because it's not really stopping it from growing. It's stopping it from translocating. So if you notice, the protein chain is translated all the way through. It's just not pushed through here. So the ribosome is still there. And only once it finishes translating that message does it dissociate. So, but when the hydrophobic sequence is there, it stops it from translocating. It's not stopping the translation. Does that answer your question? Yeah, it reads it right to the, for any of these proteins, it'll read it all the way to the stop codon and then dissociate. Great. My dog came back and listened to the end of the lecture. I think I'll post the exam grades on Friday. That's what I'm aiming for, probably Friday evening. Okay, see everybody Friday.